Hello everyone. Today we are going to learn about plant growth substances. Plant growth substances are trace chemicals that regulate plant growth and development. They are divided into plant hormones produced endogenously by plants and synthetic plant growth regulators. Plant hormones refer to some trace organic substances synthesized in the plant body, transported from their site of production to other parts, and playing a regulatory role in plant growth and development, such as auxin, gibberellin, cytokinin, abscisic acid, ethylene. Plant growth regulators refer to some synthetic substances with plant hormone activity, such as ethaphon, chlormequat, CCC, paclobutrazol, and mepiquat chloride. Now, let's start with plant hormones.
First,
Auxin. Discovery of auxin. In 1880, Darwin theorized through experiments that the tip of the coleoptile might produce a substance that, under unilateral light, would have some effect on the lower part of the coleoptile. In 1910, Jensen experimentally demonstrated that stimulation from the tip of the coleoptile could be transmitted to the lower part through agar plates. In 1914, Pull experimentally demonstrated that the curved growth of the coleoptile was caused by the uneven distribution of stimuli from its tip. In 1928, Wendt experimentally proved that the stimulus causing the curvature of the coleoptile was a chemical substance, which he named auxin. Distribution and transport of auxin. Auxin is distributed in all organs of plants, mainly concentrated in vigorously growing parts such as stem tips and root tips. Auxin transport in plants exhibits polarity, meaning it can only be transported from the morphological upper end to the lower end, and not in the opposite direction. This is called the polar transport of auxin. Physiological effects of auxin can promote growth, induce apical dominance and inhibit lateral bud development, promote rooting of cuttings, and mobilize nutrients. 2. Gibberellin. Discovery and types of gibberellin. Gibberellin was discovered during research on Rice-Bacani disease. It is a general term for a class of compounds with a gibberellin skeleton that can stimulate cell division or elongation, or both. Synthesis and transport of gibberellin. It is synthesized in young apical parts of plants, such as stem tips and root tips, and exhibits bidirectional transport. Physiological effects. Promote stem growth, promote bolting and flowering, break dormancy, promote male flower differentiation, induce parthenocarpy, forming seedless fruits. Cytokinin is a plant hormone that promotes cell division. Discovery and types. The earliest discovered was zeatin, which is synthesized in root tips and transported upward through the xylem. Natural cytokinins include free cytokinins and bound cytokinins. Physiological effects. The physiological functions of cytokinin include promoting cell division, promoting bud differentiation, promoting cell expansion, promoting lateral bud development and eliminating apical dominance, delaying leaf senescence, and breaking seed dormancy. 4. Abscisic acid. A plant hormone capable of inducing bud dormancy, leaf abscission, and inhibiting growth. Distribution and transport of abscisic acid. It is synthesized in aging leaves, root caps, etc., and exists in all organs. ABA content is higher in organs and tissues about to abscise or enter dormancy. It also increases rapidly under unfavorable environmental conditions. ABA transport is fast and nonpolar. 2 physiological effects of abscisic acid include promoting dormancy, promoting stomatal closure, enhancing stress resistance, inhibiting growth, and promoting abscission. 5. Ethylene. 1. Synthesis and transport of ethylene. Ethylene is ubiquitous in all organs of the plant and can be synthesized in living cells. It is induced by abscising and aging parts. Unfavorable environments can lead to large-scale synthesis. The content is small, and it mainly acts at the site of synthesis. 2 physiological effects of ethylene include altering plant growth habits, promoting ripening, promoting abscission, promoting flowering, and female flower differentiation. Now let's look at plant growth regulators. Plant growth regulators are synthetic organic substances that have obvious regulatory and controlling effects on plant growth and development. First, auxin analogs. 1 indole derivatives structurally similar to auxin, such as indole-propionic acid, indole-butyric acid. 2 naphthoic acid derivatives, such as alpha-naphthalene-acetic acid, sodium naphthalene acetate, naphthalene acetamide. 3 chlorophenoxy derivatives, such as 2,4-dichlorophenoxyacetic acid, 2,4,5-trichlorophenoxyacetic acid, 4-iodophyenoxyacetic acid, etc. 2. Gibberellins. GA3 is the most widely used and studied gibberellin in production. 3 acetokinins. 2 are commonly used, kinetin and 6-benzylaminopurin. 4. Ethylene-releasing agent. The commonly used ethylene-releasing agent in production is ethafon. 5. Growth inhibitors, paclobutrazol, chlormequat, diminazide, etc. Students, as agricultural practitioners, issues to pay attention to when applying plant growth regulators in production are: 1. Strictly control the concentration. 2. Hormones absolutely cannot replace fertilizer and water. 3. Pay attention to the application methods of various hormones. We must not neglect the safety of agricultural products and the sustainable development of agriculture, because we are eager to achieve success and pursue short-term interests. That concludes this section. Thank you.